Okay, good afternoon, people. So, in the last class, we had looked at different structures of mRNA, different forms of mRNA, including mRNA, tRNA, rRNA being the major ones, and of course, this hnRNA, snRNA, genetic and ribosomes. So, these were different types, and I had asked you to look at the functions. So today we will be starting with the first process, that is the DNA replication. Now, in general, you know what replication is, which is the duplication of DNA molecule to produce its identical copies. Now, DNA replication happens in S phase, that is the synthesis phase, which is a part of interphase during the cell cycle. So I hope you remember that G1 and then S phase, then G2, and then the mitosis occurs, right? So during the S phase, all of the chromosomes in the cell or all the chromatin fibers within the cell nucleus, they will get replicated. So we are going to talk about that replication process. Now, this particular DNA replication is of a type which is called a semi-conservative method. We will talk about these different types maybe immediately after this. So, why it was thought to be semi-conservative? What does it mean by semi-conservative and so on? That we'll see. So, Watson and Crick in 1953, they had suggested this simple mechanism of DNA replication. Of course, they kind of given the what is that clover leaf model of the not clover leaf model, the double helical model of the DNA structure. On the basis of that, what they found out that there can be simpler mechanism of DNA replication which can be explained. And usually this mechanism which they mentioned had breakdown of those hydrogen bond, which was the one of the weakest component or weakest bond found within those two strands of the DNA, which were connecting two nitrogenous bases. And that way, if the DNA will uncoil and then separate, and then the specific new nucleotides will go and form a chain over those separated strands. And this process is like with the, of course, with the help of those nucleotides which are complementary to each other. And that is where, that is how it happens. Now, once the nucleotides are attached by their hydrogen bonds and their sugar radicals will unite their phosphate bonds, completing the formation of the new polynucleotide chain. So that was the mechanism which was explained in brief by Watson and Crick, while they had also designed the double helical structure of the DNA. So in simplest terms, what they said was the DNA will kind of break down the hydrogen bonds. It will uncoil, it will separate, new nucleotides will be added, and then later on their phosphate and sugar will form a backbone strand, and then they will ultimately get attached to each other as a two new DNA molecule. So that was the model which was explained. Now, thus the nucleotide strand of the double helix of DNA serves as a template on which the complementary strand is built. So this is the kind of representative model which was explained by Watson and Crick. So this was the typical DNA molecule. You can see it's kind of unfolding. That means the strands are separating, and at certain time point, like whichever needs to be replicated, there will be nucleotides will start adding, and then they will form this sugar and phosphate backbone. So this method of DNA replication, where one of the DNA strand acting as a template and one strand is new, that's what is described as a semi-conservative. Semi means kind of half, and that is conserving that half molecule of DNA. So that is what is semi-conservative. Each daughter DNA molecule is a hybrid conserving one parental nucleotide chain and synthesizing the new one, and that is why it is also called as hybrid, because one strand is old or what we call as parent strand, and the other one is the new strand, and that is why this particular daughter DNA molecule is the hybrid as well as the other one as well. So that is just the general mechanism of this DNA replication, which was explained by Watson and Crick. Now, coming to these different models, like people also have thought of then different models that it can be any of these three models, what is commonly known as semi-conservative, conservative, and dispersive replication. Now, without going into description, I'll just show it to you here from this diagram. So, in case of conservative model, both the strand, I mean, these are again hypotheses, okay? These are not how it exactly happens. That is what people had thought by then, how the DNA replication could occur. So one of the model, which was conservative model, both the DNA strands, they stay intact as it is, and based on that information, there will be two different DNA strands which will be formed. So at any given situation, when one DNA molecule is replicating into two daughter DNA molecule, one will always be parental DNA molecule and the other one will be a newly formed molecule. So that is what is conservative. In case of semi-conservative, the two of the DNA strands, they will separate and they will maintain, or they will make the new DNA strand on the respective parent strand. So they will have one new strand and one parent strand. So that way it is kind of semi-conservative. So again, at any given situation, whenever the DNA replication happens, it will always have one DNA strand from the parent and the other DNA strand from which is newly formed. So that is semi-conservative. And dispersive, which involves like kind of fractions of both the fragments. So first patch from the first DNA fragment or DNA strand, then the second portion will be from the next strand and so on. So that is what is dispersive method. Later on, scientists went ahead and tried to study which method could be possible, and obviously they found it is the semi-conservative method, which occurs in particular DNA replication process. Okay, any question here so far? I hope this is very simple to understand.
So is semi-conservative the most common, or is it the only thing that happens?
Sorry, in semi-conservative?
Is it the most common out of these three, or that's the only one that happens and
That's
the others are just
one that happens. Until and unless it's mistake, then the conservative might occur. But in general, all of the cells, they have the semi-conservative method of DNA replication. Okay, now how to prove this semi-conservative DNA replication? So then there were quite a few experiments to prove that the replication happens in the form of semi-conservation and not as a whole like conservative or dispersive way. So this particular fact was established first in 1958, by Meselson and Stahl. This is very well known as the Stahl's experiment, but in general it is Meselson and Stahl's experiment. But if you just find somewhere like explain what is Stahl's experiment, that means the same experiment. So they studied this bacteria called as Escherichia coli. Now the thing that they did in this was they had this N15 isotope of the nitrogen, and then there was normal N14 isotope which is normally present, and these can be distinguished based on their densities by the centrifugation in the cesium chloride. Cesium chloride is kind of a material which is very widely used in the centrifugal processes in the biological experiment, because it has a certain density, and based on that density the molecules they separate in the cesium chloride solution. And if you add along with it something, let's say for example these DNA molecules, so their separation will also be controlled like in the form of layers, and that is why this cesium chloride is used. If you want to read more about it, I guess this is the reference that I have attached here. Yeah, cesium chloride density gradient centrifugation. So this process really explains. So if you see in this kind of diagram, a small diagram that is coming up, there are two DNA layers. So this white patch is one DNA molecule, this other patch is another DNA molecule. Obviously the heavier one will settle at one place, and the lighter one, based on the density, it will settle at one place. But again, we need not go into much details of it. If you want to read, the information is there. So what these Stahl and Meselson did is they cultured this Escherichia coli in a medium which contained heavy isotope of nitrogen, that is N50, in the form of ammonium chloride. So most of the culture medium in general, they contain ammonium chloride either as a source of nitrogen or also as a buffer system. That is where the most of the bacteria or cells they take the nitrogen from the culture medium, that is the nutrient medium. So once these E. coli, they are grown with the heavy nitrogen, that is N50, and that too for many generations, this produced population of E. coli bacteria with both the strands of their DNA containing N50, and which was the heavier than the normal N14 isotope of nitrogen. So
did they do it in the same culture or in two different cultures?
Only one culture. So what they did is they took E. coli bacteria, they put it in the culture medium, and the culture medium was totally made up of this N14. So only N14 which will be there will be the original E. coli bacteria having their DNA molecule, right? So that will be the only nitrogen which will be N14 originally. But as and when the replication happens, they will keep replacing it with the N15, which was the heavier molecule. So there was only one culture. Okay, so this.
So in one culture, you mean that in one culture there was only one type of nitrogen. So there were two cultures with one type of nitrogen each, or in one culture they supplied both the types of nitrogen. So a bacteria could get N14 or 15.
They only used N15. So at the start, obviously, whenever we had a culture of E. coli or Escherichia coli, it must have been grown in a normal N14 isotope, right? That was the ammonium chloride which was used earlier. So from that, you take small amount of bacteria and you culture it in the heavier form of ammonium chloride, that is heavier form of nitrogen ultimately, N15, and then you grow those bacteria only in this heavy nitrogen containing culture medium.
Okay, so there was no N14, there was only N15.
In the culture medium, yes, but during the when we take out that E. coli culture, a part of it to put it in the heavy nitrogen culture, there will be a little amount of DNA present in those E. coli cells. That will be the only N14 in that.
Okay, yeah. So inside the E. coli there's N14, but in the culture there's N15.
Correct.
Okay.
And now they so what happened is, after few generations, all of the E. coli will have only N15 because the replication will keep happening. So what that is what is the process. No, not this one. Where is it? This is no, not this one as well. So let me show you here itself.
Sir, that previous slide, it said something about E. coli.
Which slide?
The one that you like, the one that you skipped. No, the slide. There was a pink color bottle on top of that. It said E. coli. There was a diagram, a big diagram.
Oh, okay. This one.
Yeah.
Yeah. But I'll come to this a little later, because this may not explain the process that I want to say. So what I wanted to say is here. Let's say, for example, this is the semi-conservative method. Now, at first generation, these, let's say these gray ones, these are the what we say N15 or rather originally they were N40, right? All the nucleotides, they had N40. Now we put it in the culture medium containing N50. So that means this blue one will be N15, again half of, so an N14 will remain half. And over the time, when let's say 1000 or maybe many more generation has been crossed, the amount of these will be still only 2 molecules of DNA, and all the new ones will all have this blue or N15 nitrogen, right? I hope you are understanding, because when this molecule will replicate, again this will separate, so it will have 2 from this and 2 from this. So in 2 from this gray or N14 containing nucleotide, it will only have 1 molecule until the end, because the original molecules were only 2, and when they replicate even until end, this original N14 molecule will be 2, but the number of N15 molecules or N15 containing DNA molecules will be quite high. So it will be somewhere like, let's say there are 100 molecules formed of this DNA, there will be 98 of N15 containing DNA molecule and only 2 of this parent strand. Or even let's say there are 1000 DNA molecule formed. In that also there will be only 2 parent strand, because which will have N14, but rest all 998 will have this new strand containing N15 nucleotide. So that is what is the meaning of not this of this statement. Where is this? Several generation. Yeah, so they cultured the E. coli in a medium constituting heavy heavy nitrogen over many generations, which produced the population of E. coli with both the strands of their DNA containing N50, and which was the heaviest molecule. Now, what they did after this is, now they separated the E. coli bacteria from that culture, and they put this bacteria into normal N14 culture medium, right? N14 ammonium chloride containing culture medium. So what happened there was, again the bacteria will start dividing. So first sample from this culture was taken after replacement, that is 20 minutes. Now this 20 minutes is coming from the doubling time or generation time of this E. coli. E. coli doubles its copies in 20 minutes. So every 20 minutes, if it is 1 bacteria, it will become 2. At 40 minutes, it will become 2, 4. Then at 60 minutes, it will become 8, and so on. So it goes on exponentially increasing, like exactly double of it. So that way, this like after changing the medium, then you take out the sample after 20 minutes, and what was found that 1 strand was heavier. So again, what was found, as in, again you take out the E. coli bacteria along with the culture medium, you separate the E. coli, you digest those sample, extract the DNA, and then you let it undergo the centrifugation process in the cesium chloride solution, and in that what you will find is those 2 bands in a particular tube in which the centrifugation was done. One which is heavier, that is N50, which is coming from the parent strand. Now in this case, all the parent strand, they have been changed to N50 because of the several generations of their culture growing with N50, and now we have replaced with N14. So some of the new strands that are being forming, they will have this N14, and which indicated the semi-conservative method. Now I can go back to that diagram. So here is the start. So N15 containing medium, which is shown here as pink. Then DNA is extracted from these cells after 20 minutes or after, let's say, replication cycle. or even at the start, where you can take it as a control, you can take out some sample. So you centrifuge it, you will see all of the DNA with N15 heavy DNA, because both these strands will have N15. That is why it is shown as double. So heavy DNA, and you will see a kind of smear. I hope you are able to see this. So you will see a kind of smear as a whole thing. This is the all of it is N15, that is the heaviest band, and this is again confirmed with some okay, I can't really, I guess let me open it in a new tab. So this is Meselson Stahl and Stahl experiment. It is this one. So I can just zoom in the diagram here at least. There I can't zoom in. Oh, the clarity goes for a toss here then. Okay, very bad. Or I can just try a little. No, it's impossible. Sorry. Okay, let me go back here then. To Scheiße. Why is this not going away? Okay, let me try to do it here, because I guess this upper part may not be visible. Am I right? It must be getting blocked. Okay, now it should. Is it visible? Are these label visible? DNA composition.
Yeah.
Okay. So what I wanted to show is these are the photographs of the DNA bands. I don't know if you are able to imagine the whole of the process. Now you might wonder, where are these photographs coming from? So usually you take out this layer that you have got in this solution, and you load it into a gel-like structure, and you let it run over time. So the molecules with the same weight, same molecular weight, they will run at a certain as in they will go at a certain speed through the gel. And then what you get is a band of this, all of the DNA molecule together. And that is what is the densitometric scans. Again, these are the specific densitometers where you just keep the sample and it will give you the density. It is like a direct automatic measurement. So you get these one band. Now, when you take the sample after 20 minutes, now you see the medium, which was N15, has been changed to the regular medium. You let it grow for 20 minutes, which is one generation time. And then you take out some sample, again extract the DNA. So here what you will have is one DNA strand will be of the parent molecule, that is N15, and the other DNA molecule will be of N14, which is newly formed DNA strand, and that will be taking N14 from this ammonium chloride present in the culture medium. So that way it is saying half of it is new, which is N14, and half of it is parent, which was N15, coming from this pink culture. And there you will see a lighter band of this same what we say, this N15 was visible as a dark band. That means it was quite a lot in amount, and you can see the intensity of this band has reduced to half, and that is why you see that only N15 band, but it has reduced to half of its intensity, and same you will get in the densitometric scans. Now again, because we don't have Y scale here, you might wonder why this graph or peak looks like exactly similar, but ideally if you look carefully, this peak is little smaller than this peak, right? So ideally that way, yes.
Why did you say that it grew lighter?
Because it is lighter.
No, like it's lighter in weight, but like why would the photograph show be less dark?
Because less amount of N15, no? Here means the DNA
So
which has
it will just be displaced to the right or to the left, right?
no.
Lighter.
Yeah, sir. Because like it's a photograph, right? And it's black and white. So I mean, if you keep a balloon and if you keep a brick and you take a photograph of the shadow will still be black.
No. So that is not how these experiments are done. So usually when we do these experiments, let me show you that. I don't know if I'll be able to show it. ethidium bromide agarogel electrophoresis. Yeah, so these are the perfect images. Now if you look here, so this or maybe this one, this is little larger. So what you have is the ladder here, that these are the markers. That means individual DNA band is of specific molecular weight. So this is usually 10,000 base pair, then 5,000, then 3,000, 1,000, and so on. So last one is usually 100 base pair and so on. So that way we have a ladder. So you let your sample run alongside it. So you you load the ladder, you load your samples, whatever they are. So they will run and they will separate the DNA molecule based on their molecular weight. So now here itself you see a kind of sample that this amount itself is too high. That's why this is bright band. This is the brightest band. Now this picture is taken in one go, right? This whole of this picture is taken at one go. So if I cut this band here and if I show it to you, and if I cut this band separately and show it to you, we cannot say that because you adjusted the brightness, that's why it is bright or light. Yeah, so that is how the experiments are done. What you are seeing here is exactly the same, a small patch.
So
Yes,
how dark or how light it is depends on the amount of it, right?
yes. So that is when I said, like, if you look at this band, the uppermost band, which is thicker and which is more darker, and this band is lighter compared to this, and it's less thicker. So obviously the amount of N15 in this DNA is not there. Ideally, what should have been seen is.
You said that it depends on the amount, the thickness of the band.
Sorry.
So it doesn't depend on the amount.
It does depend on the amount, of course.
So I mean, any could have been more, right? Like N15, let's say there are N number of N15 DNA. And when you put it in the other medium, it doubles because it replicates itself. So there are 2N. So there's more.
No, so again, for that, what we do is after DNA extraction, you measure the amount of DNA in that particular sample, and you take equivalent amount of DNA for each sample, and even the volume of the sample that is loaded in these samples, they are equal. So you make sure of that. You just check. So the only variable that is left is the nitrogen that is present, which is N15 or N14. So you
are saying that molecular mass makes them larger and denser.
Correct.
So, but like these ladders, they don't have a pattern, right? I mean, if they are taken at the same time. So if we say that the thing that's heavier will move down slowly, or it'll move down faster, any which way. In the first, the heavier thing is on top. In the second, the heavier thing is in the middle, and in the third, the heavier thing is at the bottom.
No. This is not the heavier here. It's just the amount of DNA of this particular type, of this particular molecular weight is higher.
So, but how do you know that? Like all amounts are taken equally, right?
Correct. But then that is when I'm saying, let's say in this case, when N15 was the only one, that means whole amount of DNA strand had this N15, and that is why all of the DNA molecules, they will run at the same speed. They will all accumulate at one place, and then they are running at the same time forward in the gel.
So I had a question. So like a heavier particle will run faster or slower?
Heavier particles will be slow.
Okay. Yeah.
So that is when I said you load the sample on the top. So heavier will travel. Let's say this is gel is run for 1 hour. Heavier particles will only travel this much distance. Yeah, these are all heavy ones. These one will be little heavy ones. These one will be lighter ones, and this will be the lightest one in terms of weight.
Yes, but that doesn't have anything to do with how bright they appear or how thick they are, right?
Sorry, that doesn't have
their heaviness, their atomic weight. That doesn't have anything to do with like how bright they appear on the gel or how thick they are, right? It just depends on the speed.
No, I still did not get the question.
So like when you want to see their molecular weight, in the gel,
Hmm.
it only matters, only the speed matters, right? That the heavier thing will go slower. The thickness of the band that you see or the brightness of the band doesn't matter, right?
It does matter a lot.
So, but then
That indicates the amount of DNA that is present. Like for example, here you see both the bands, they are of same molecular weight. But in this sample, this band or this DNA molecule had a very less amount. But the same molecular weight in the other sample has lot of this particular DNA, and this is less and this is more.
yeah, so the thickness and like the thickness and brightness depends on the number of particles that are there.
Correct.
Yeah, okay. So in the previous case,
In this case,
so the first one shouldn't be thicker, or like it may or may not be thicker and like darker than the second one.
why?
Because the number of particles can be more in either. So
No,
it's like,
that is when I said you take exactly same amount of sample.
yes, if you take the same amount of sample, then they should have equal thickness and equal darkness, right? I mean,
You mean first and second case?
yes, sir.
That is what I am trying to tell you. In this case, N14 is separated from N15. What you see here is only N15. You don't see here. You can probably see a very faint band here, right?
Oh, so from the.
So that is what I am trying to tell you. N15, because of its heavy molecular weight, it will run a little slow. And N14, because of the lighter weight, it will run a little faster. So you will see two separate bands.
Okay, so the DNA, they actually broke the strands of the DNA into two.
It ultimately breaks up. While you run it on a sample, you denature the DNA. So you separate the strands ultimately. And that is what runs in the double helix. So I guess, let me show you.
But if you see the last diagram, then that wouldn't make sense, right? If the bands actually separate and run away, then the middle
because the amount of N15 there is almost negligible. Like I said, let's say there are 1,000 molecules of the total DNA. Only 2 of them will have N15, because at the end, only 2 parent molecule will be passed on to the next generation. They will not have more than that.
Sir, no, that's not what I'm saying. If you see the middle band, the N15 and
this one.
That won't exist, right?
Why?
If the strands actually separate and go down, it will only be 2 bands of N15 and N14.
So that is what this is, no?
No. The middle band, then there are 3 bands, right?
No, this white is not the band.
So no, the last diagram, bottom.
This one, huh?
So the middle band, they've labeled it as intermediate, N15 and 14, right?
No, okay, listen to me now very carefully. This band is on N15, this is at this length. Yeah, this band has reduced its intensity. It's actually on one line, but because this has less intensity, it looks like little like towards your left side. Okay, but they are of the same intensity. Both the bands, they are for N50. This third one is also N50. This one is N40. Okay, and this one is only N40. There is no N15 here. So what they are trying to say here is N15 was originally there. As and when the generation time increase, the N14 amount is increasing and N15 amount is decreasing completely. So that is the conclusion of these photographs. Did you get this?
Like, yeah, I got the experiment, sir. But yeah, so the black band in the second experiment, is that N14 or 15?
This one is N15.
And N14 is to its right.
N14 is
to its right.
Towards left.
Okay, it's towards its left.
This one is N14. This one is N14. These 3, 1, 2, and this third one. These are N50.
In the third band, there are 3 bands. It seems like there are 3 bands.
Why? Where are 3 bands? I can only see 2.
So there's one black, and there's one green, gray and this one black again.
This is not the band, Pratham.
Okay, that's just space.
Smear. So that is what I was trying to show it to you. Even if you look here, you can see there is quite a lot of smear. You see this line here?
Yes, sir.
Yeah. So that's a smear. Even if you look closely here, you can see a very faint band. Yeah.
Yes.
Right. So that's something like when you have the DNA sample, some of the DNA molecules may be broken into half, maybe broken into 3/4 or whatsoever amount, right? You may not have all DNA molecules always intact, right? That is the sampling processing problem, right? Or it can be room temperature, which is little higher, so DNA gets degraded, or some process while the DNA extraction was done a little wrong. So that is all these processes, or the DNA is out of the cell, that stress. All these factors, they contribute to the breakdown of DNA. And that is what you see a little bit of smear. So more accurate the band or more sharpness of the band. For example, here you see, right? You can miss, I don't know, like if you are able to see it clearly, but quite clearly there are these two bands. Here there is little bit of smear, but in this band there is almost nothing, right?
Yes.
For example, here also very clean bands, right? So that depends on how the sampling was done. So always you will see some kind of smear until and unless the sample was done like as perfectly as possible. So that is why you might see that as a third band, but it is not a third band.
Yeah, so like in the second also there is presence of N14 and N15, but why have they only shown N15 and not N14?
Because the amount of DNA that will be present in these samples will be relatively very less.
So, but it has to be 50-50, right?
It has to be 50-50, but still it may not be so visible here. And that is when I say ideally there should have been a faint band here.
So, but it will be a band of equal length and equal thickness because the amount is the same.
Not necessarily, Pratham, because this culture may not have all the bacteria in 20 minutes, which would have gone to the gone through one cycle of replication. That is an assumption that 20 minutes is the doubling time. That does not mean that within that 20 minutes, all the bacteria they would have processed or finished their DNA replication. There can be a little delay in the cell cycle, or there can be some cells would have started by the mid process, right? So considering all these processes, like not all these cells, they are at the same cell cycle stage at any given point. So that is why you may not be able to see N14 band here so distinctly.
So you're, but so like even if that happens, so yeah, so even if the time for one cycle is 20 minutes, and if you see the amount of E. coli that's there, and if you see the amount cycle, the band should still be there. I mean, it'll only be like 2% dimmer or something. I mean, the more number of samples you take, the more the probability of getting the right increases, right?
No, I didn't get it again.
Like, so there might be a probability that some won't be able to do because won't be able to undergo cell cycle because of the factors that you said, but most of them will still, right?
Most of them will still, but then in that case, that is what I'm trying to say. Let's say there are 1,000 cells, out of which 300 cells they are not able to undergo what we say cell cycle division, right? 700 they have gone through the cell cycle division. Now from that 700, all of them they will have half molecule of N15, half molecule of N14, right? N15 half, N14 half, right? And the remaining 300 will have all of it as N15. Now if you calculate the total amount of N15, it will be quite a lot, which will be like ideally 700 plus 700. 700 will be N15, 700 will be N14, that is half, and plus 300 plus 300, because both of the DNA strand in that non-dividing cells will be of N15, that is from the original culture of N15, correct? So that way, if you look at the amount of DNA in this case, will be all N15 majorly, like 700 plus 600, 1300 compared to 500. Compared to 700, which is of N14, which is almost double. So that is when you don't.
Values are an assumption. Well, so the values
No,
are
values are not assumption. If we consider there are these many number of molecules, that is exactly how it would be. It cannot be an assumption because DNA molecule has 2 strands.
No, like an assumption 700 and 300.
Correct. But usually that is the case. Almost half of the cell, I still took more on one side. Almost half of the cells, they are in different cell cycle zones compared to the fast growing cells. So that is the usual case of the cell cycle division.
Okay, so well, if we take that, so you're saying that we are missing a light band here in the diagram.
Yeah.
Okay. Okay, fine. Yeah, I got that.
And like I said, ideally there should be a lighter band shown here.
And you're saying, and like that's why the band that's supposed to be lighter there keeps on increasing in thickness as the
N14 is increasing. Yeah. So lighter, then thicker and thickest. So that should be the band here, like this, N14. And N15 is disappearing. N14 is like kind of increasing in thickness. So that is the perfect.
If you see the last diagram, then they've labeled 3 bands. So but they should have labeled only 2, and they haven't labeled strands. They've labeled DNA.
They have labeled the position of the bands where they should appear.
Yes, sir. Where they are appearing and how much their amount is, right? They've labeled that. So the stand shouldn't be there, right? Then because that's not
so that is when, like in some of the experiments, when you don't denature the DNA, that is what you will get here. Right? Ideally, you should hold a scale and then look at which band is falling in this line of this, which line is falling in this. And this could be the band which is of N14 and 15 together. So again, like we assume so many things. We don't know how these bands were created in these experiments. But biologically, when we speak of it, this band could be of these 2 things, N14 and N15 together. So which should ideally appear as an average length.
I don't think they're separating it into 2 separate strands here. They're putting them as it is, as DNA molecules. So the first
No, they are not separating strand. When you prepare the sample, it ultimately gets denatured. Sample preparing,
denatured, then it might move a bit to this side, it might get smeared and all that. But I think they're trying to keep it as one DNA molecule, not separate it into 2 strands.
Correct, you are right. That is how it happens.
Yeah,
in the sample, but when you process the sample, when you add certain buffers to that sample to load the sample, this DNA ultimately gets separated.
Yeah, so I think the densitometric scans clear it out because all of them are in the second one, all of them are in the middle. For the third ones, half of them are intermediate, half of them are light, and for the last one, most of them are light.
Yeah, but again, in this case also, the amount should be very less.
Yeah, they've exactly.
That means if it is showing here N15 bands, then it should show N14 band here also, right? Because these 2 situations are exactly opposite to each other. Right, this is complete N15, then it starts shifting to N14, but then there has to be a little peak of N14 here.
No, sir. Like what Akshay is saying is that because there's one DNA, there'll only be one band per DNA. So the amount gets denatured. That's they aren't considering that. That's negligible. Otherwise, they wouldn't have shown this type of diagram. They would have shown a bit different diagram. Because in the first two cases, there's only one type of DNA. In the second, there are two types. So there are two peaks. And in the third, there are three types. So there are two peaks, and well, they've labeled one more.
Yeah, right.
Also, sir, I have another question. What are they trying to do with this experiment?
Okay, so they are trying to say that as and when the DNA replication happens in a particular cell, half of the DNA is of the parent and half of the DNA is newly formed. So that is what they are
It will
trying
give
to.
the same results even if you take the other methods of this thing.
Other types of cells.
Yeah, so where both the parent strands go to the same cell and where there's distribution, both of those also will give same results.
Yeah, any cell in general should give the same result.
So the slide said that it was trying to show that there was separation, the
Conservation.
Yeah, semi-conservation.
Correct.
No, what Akshay is saying is that he's saying that any other method, like the conservative or the dispersive method, you get the same result.
No, they will not give any. That is what I'm trying to say.
Yeah,
Later on.
Because if you take the completely conservative thing, then half of them will have it, right? Because the other half will be completely new one.
But that is what they are trying to prove here by making this N14 and N15, no?
Oh, Akshay, then there in the second diagram, there will be two peaks because one will be N14, N14, and one will be N15, N15.
So exactly, there will be completely separate bands, different bands rather. The band pattern will be different.
So the second diagram will be like the third diagram. And the max peaks can only be two, but there are three in the last one.
Okay, yeah, fine.
Okay, so I guess enough of discussion about this. So I guess you understood whole of the experiment. So similarly, what they had done is 20 minutes they have taken one sample, then not this. 40 minutes they have taken another sample, and 60 minutes they have taken another sample, which is third generation. So that's when the 70% DNA was normal and the 25% was the hybrid of N14 and N15. So that is the general like outline of this Meselson and Stahl experiment. This video will explain everything again, so you can watch it on your own whenever you have time. This is like a beautiful animated video of the same experiment. So please do watch that. How much time do we have? It's 26. We may not be able to finish this experiment. So there is this another experiment. Okay, I guess let's try it. If at all it is remaining. No, we will have. Okay, let's try it. So there was this another guy whose name was J.H. Taylor, not Tyler, sorry. So he demonstrated again the same experiment, but in a specific root tip cells of this Visia faba. This is one type of plant. You can Google it out through the method of autoradiography. So that means he took the radioactive thymidine and he incorporated in the DNA of the cells. Again, incorporated means he allowed those particular cells to grow in the medium which contained radioactive thymidine. So that way the cells were grown. So obviously all the cells will have this radioactive thymidine and so on. So that is the usual way of doing these things. So the outline of the chromosome labeled with the radioactive thymidine appeared on the photographic film. So earlier there was no radioactive thymidine. Later on, as and when the medium containing radiothymidine was used, it started appearing in the photographic film. So when these tips with the labeled chromosome, labeled as in radioactively labeled chromosome, they were transferred to the unlabeled medium. So that means after a certain amount of divisions of the cell cycle, These were again transferred to the regular thymidine containing medium, which was unlabeled, and it contained colchicine. Now colchicine is specifically used to arrest the cell at a particular cell cycle stage. So that means whatever cell cycle those cells will be at, they will arrest those cells in that particular phase. It can be mostly colchicine is used in the what we call as metaphase blocker. So that means once the cell reaches to the metaphase, this colchicine will act onto the spindle polymerization, so that mitotic spindle assembly is kind of disturbed and the cell goes to that fixed stage. So it does not go to the anaphase and telophase further anymore. So all these cells will be blocked at this metaphase using colchicine, and then they are studied for the radioactivity. So in this, what was observed was the chromosome of the first generation, the radioactivity was found to be uniformly distributed in both the chromatid. That means when the cells were grown in the radioactive thymidine, obviously all the cells or all the chromatids will be with the radioactive thymidine. And in the chromosomes of the second generation, only one of the two chromatids in each chromosome was radioactive. That means half of it was radioactive, half of it was normally labeled or unlabeled, right? So that is what is the experiment says, and this is what is the shown in the diagram. Like if these are the chromatids, if these are the chromatids present with the normal thymidine, and you have grown these cells in culture medium which contains radioactive thymidine, then you take out these cells, you put it into the normal medium which has normal thymidine. Obviously you will start getting half of the cells which will have unlabeled thymidine and half of the chromatid, because half will be new from the parent cell and half will be newly formed. That is what they are trying to say. And the similar experiment which even Meselson and Stahl they had done, but they have just used the heavier form of the isotope, and that was not radioactive. This was specific to radioactivity, and the same thing was proven again with the help of this root tip cells of the plant, Viscia fava. Okay, any question here? I guess you understood this experiment, right? This has like similar explanation to the earlier experiment. Only the difference, like I said, is this part being the radioactive thymidine, and in earlier case it was just heavier isotope of nitrogen. And most important like thing for this experiment is the colchicine treatment. Again, there is this article that I have attached to this, which talks about the mechanism of colchicine in the cell cycle. So this particular cell cycle will be arrested at the metaphase, and then it will block the spindle polymerization. So ultimately cell cannot finish the cell cycle division and it will stay in that stage. Excuse me, I'm still in the class. I'm still in the class, I said. So please do not make noise. Okay, do you have any question? Pratham, is that part clear? The second experiment?
Yes, sir. So in the third diagram, were there 3 strands, like bands?
In the third diagram, so you mean this one?
No. The other one. The one we spent so long on, sir.
Oh, this one?
Yeah. In the third stage,
This
there are 3 bands, right?
No, there are only 2 bands.
No, but if you look below, the gray band in both the experiments.
See, we don't call it a band until and unless it is of some intensity. So when we do these photographs of this gel electrophoresis thing, we should be able to calculate the intensity of that, right? and that is when like I showed you this diagram, right? So these ones they have specific amount, also specific intensity also. For example, if you look at this band, which is ideally a merger of two bands, right? And then intensity of this band is also higher, and the intensity goes on decreasing because it is of a particular amount and of a particular molecular weight. So that is when you compare, let's say this was 1 molar of DNA, and this is of the same intensity and same molecular weight, so they will run parallel at the same time. So you can assume this band slightly lower, so the molecular weight of this DNA is slightly less, but intensity wise, if you look, they look similar. So you can approximately say this amount of DNA within this gel is 1 molar, and the molecular weight can be slightly less than this. So that is how you always compare. So that is why we always run this ladder, or what we also call as DNA marker. Okay, is that clear now?
Well, so that's not exactly what I was asking.
Okay.
The middle layer. So, is there something there, or is there nothing there?
In this middle layer?
Yes.
No, there is nothing.
Okay. Yeah. So,
So that is why I was trying to say as well, when there is, until and unless there is a certain band of certain intensity, we do not consider it as a band. We always consider it as a smear. Like for example, if you look here, this gray part, some people can call it a band, but it is not a band. It is just a smear.
okay.
Yeah. Okay, let me stop here. These people are here. I will see you people in the next class tomorrow. Yeah. Bye bye.
Hi, sir.